All right, thank you very much, Gemma, for the lovely introduction. So I'm going to talk to you about how we can use longitudinal imaging to study traits in mouse models of neurodegenerative disease today. And I think, again, like I mentioned at the top of this, our goal with doing animal imaging isn't necessarily to study the animal itself, although it's a helpful tool with which to understand brain structure and function or disease, but it's really to understand something about human disease or human behavior. And if you go to any number of talks at this conference, you'll see something at the intersection of this, where you want to look at brain architecture, something longitudinal in lifespan context, some about genomics or genes or genetics, some about cell types and transcriptomics, and we try to infer all this information from different sources, thinking, if you consider the Allen Brain Atlas as one of the canonical sources of information that we use, and try to build up really sophisticated statistical models to actually link our phenotypes together. Of course, in an animal model, a lot of this is a lot easier to do. It has obvious advantages, of course, compared to the human, and imaging offers a lot of ways in which we can kind of bridge the gap between what we see in human disease and human phenotypes and what we can do experimentally in animal models. So we saw a beautiful talk earlier today about how we can get sophisticated assays of behavior using touchscreen methods, and I'll talk a little bit about that and how we're using that in our lab. The lifespan context becomes a much easier thing to assay because a mouse goes from being born to being adult in 90 days, right? And so if you want to learn something about how a mouse develops, grows, ages under certain circumstances, a PhD student can can work for 90 days, I should say, in order to make that happen, and then some. The genetics, obviously, there's exquisite control experimentally on that. Histology is something that you can do in an animal model much more readily than you can in a human, and of course the transcriptomics become accessible. So all the work I'm going to talk to you about today was done at the Douglas Research Center, which is the main psychiatric institute associated with McGill. We're out by the St. Lawrence River. We have a lot of green space. It's a lovely place to come visit, especially in the summer. Play frisbee on the lawn, which we do. And then sometimes we use some scanning on the 7T Bruker biospec that you see here. And that's kind of the main workhorse. So it's getting a little long in the tooth now, but it does the job for what it is that we want to do. And the story I want to tell to you today has to do with synucleinopathies. And so, the synucleinopathy people know most about probably is Parkinson's disease. And we know that the hallmark symptoms are stoop posture, tremor, gait freezing, shuffling, and so on. And that kind of is amongst a constellation of other diseases where the accumulation of alpha-synuclein in the brain impacts structure and function and ultimately behavior. And so if you look back to the classical literature by Brack and Brack, looking at how alpha-synuclein was characterized in the brain, there are several theories about where synuclein comes from in the brain and how it gets to the brain. So some people, there's controversies on whether it comes from the gut, whether it originates in the brainstem, or whether it comes from the olfactory bulbs, or some combination of all three. But what's been observed time and time again, and that's what this kind of panel in the middle is trying to show, is that as synuclein spreads and it accumulates rapidly across different parts of the brain, the severity of the disease, whether it's Parkinson's or otherwise, gets worse, right? And we know that it starts in one place, and if you were to look at different brains at different periods, we can see the spreading of that disease, of that pathology in a somewhat stereotyped way. So alpha-synuclein is a protein that the brain, like in all neurodegenerative disorders, it's not something that's foreign to the brain, it's something that your brain produces and somehow can no longer cope with. And so what we do know about it is that it has probably something to do with synaptic function. Most talks about alpha-synuclein will start and end there. But what we do know is when it misfolds abnormally, it can create Lewy bodies and create Lewy neurites, which under enough concentration can seed and cause neurodegeneration. And so this has led to the prion-like transmission hypothesis. And cue Jake Vogel walking in at the perfect time. If you saw his talk yesterday, he spoke elegantly about this in his presentation yesterday. And one of the really nice observations that was made about this prion-like transmission hypothesis was after a clinical trial that was done where individuals received stem cells transplantations in the substantia nigra, where pathology generally grows, that didn't really work. But upon autopsy, what was observed was that alpha-synuclein propagated into these new neurons that were grafted into the substantia nigra, suggesting that they propagated somehow through the environment that they were in. This led to a series of other experiments where alpha-synuclein was added in vitro, in dish, and raised toxicity, and started, when it was introduced to neurons, it started kind of getting into neurons as well. And finally, this led to a series of experiments by Kelvin Luck characterizing if the introduction of alpha-synuclein into brains of animal models and how it might propagate throughout the brain. And this forms the basis of the experiments that we're doing. So we've been doing this work in a mouse called the M83 model. Basically, it hosts a transgene, the SNCA transgene, which basically is observed in the familial form of Parkinson's disease and is known to increase the levels of alpha-synuclein in the body and in the brain. And this mouse has a propensity to create it at abnormal rates, and which is what we think triggers misfolding of this protein. But what you can do, and if you think about this kind of prion-like spreading hypothesis, is that if indeed there is this prion-like spreading that's happening, if you were to seed or in some way dope the animal in a specific spot, and you add what we call preformed fibrils of alpha-synuclein, so almost like a light version of the alpha-synuclein in a known place, the dorsal striatum in our case, which we can inject, you can kind of sit back and watch what happens to the animals as you do that, right? And you would expect that if there's a prion-like spreading after you introduce that alpha-synuclein into the brain, that after some time you would get more alpha-synuclein in other parts of the brain, which would cause neurodegeneration. And so what happens if you do that in this animal? So on the bottom is you see an animal that's been injected, is that right? And on the top you see one that has been uninjected. When you see them side by side, you'll see that the injected animal is always going to be the smaller one, so it's the easiest one to keep track of, which is one of the hallmarks of this surgery working. You'll see some gate freezing, like you see right there. It doesn't have a fluid emotion. It kind of sits out by the side of the cage, and then when you lift up the bedding, which is done here, the animal that's uninfected can rear, can go trying to seek out novelty, while the other animal kind of is just trying to keep ambling along. And so this is how you know the animal has is fully symptomatic, is close to reaching its endpoint, simply because the impact of the disease to become too great for it to become mobile. And so I'm going to describe a few experiments that were done by a very impressive PhD student who's worked on this long and hard in my lab, Stephanie Tullo. She learned how to do all the surgeries when we didn't know how to do them, did all the behaviors, brought all this to our lab de novo over the course of several years. And most of the experiments I'll show you today will follow this design, where the days aren't terribly important, but we do some sort of longitudinal imaging. We image before we inject the animal with alpha-synuclein, and then we assay the animal several time points. And then we expect the animal to be sick starting around 90 days after the injection and reaching its final time point at around 120 days or thereabouts. We do this we image the animal all in the 7T Bruker, like I showed you before. And of course, there's a big difference between, say, the human brain, where we expect 1 millimeter voxels. So we make up for that 1,000-fold difference in brain size by having 0.1 millimeter or 100 micron voxels isotrophic in our animal models. We use a standard deformation-based morphometry pipeline to analyze these animals. So basically, we look at within subject change over time using a subject-wise averaging. If you think of like an ants model build, for example, it's the same underlying code that we use. And then we bring all that into a common space to create a population average where we do our stats. And so when we do this is the type of canonical atrophy pattern that we get. If you haven't seen much in terms of mouse brains, on the left you see the cerebellum, or the back of the brain, or close to the cerebellum, moving forward towards the front of the brain on the right-hand side. And even though we inject in the dorsal striatum and on the right-hand side, we get bilateral atrophy in the animals that have been injected, and we get regions that are kind of interesting in the context of movement disorders and synucleinopathy, including periaqueductal gray, the substantia nigra, so it descends into the substantia nigra, caudiputamen, the accumbens, and the frontal cortex. And we can assay this using kind of a network-type propagation model, so in this case, or even like a very simple matrix factorization model, like non-negative matrix factorization, where you can get subject-specific loading on this, and we see more of this atrophy pattern in the injected animals than not. And this very much is in line with what we see in humans. And so this is beautiful work done by Yashar Zagami, who's now a professor at the Douglas Research Center as well, showing that similar regions atrophy to the ones we see in animal models, giving us some sense of translatability of this model. Okay, well, the advantage here is that we have mice, and we have an MRI signature, and we can look at things that might be related to atrophy. So for example, here we see astrocyte load and microglial load are the first top two panels, and that's increased, so downstream players in neurodegeneration and inflammation that might be playing a role in the neurodegenerative phenotype that we see, and increased alpha-synuclein concentration right near the place that we see. So that's the bottom panel that you see there. The plots on the right kind of tell you the differences between groups, the injected and the uninjected groups, suggesting that this did the job. If you look at another region that's relatively unaffected as a control region in the brain, you don't see that difference at all, right? So suggesting that this is really specific and honing on neurodegenerative processes that we may have. The other thing we can do, there we go, is we use the touchscreen that was so elegantly described earlier today. And just for those of you who missed it, basically a mouse is taught to differentiate between two different stimuli. When it gets it right, it goes in the back and gets a milkshake reward. The type of milkshake you use is so important. This is one of the things I learned. We have to import it from a neighboring province just to get the right type, or else this whole thing falls apart. Absolutely true story, and it took us way too long to troubleshoot that. Basically, we changed the rules to assay the cognitive flexibility, and so that's a reversal. And these are the trials that you see for it to get to understand the new rules. And what you see is that after 50, 60 days, you start seeing a deficit in these animals that are injected relative to the uninjected animals. And those injected animals, after 90 days, are performing at chance. And that's what that pink curve is showing you relative to the blue curve. So if the prion hypothesis is true, we think that synuclein clearing should reverse neurodegeneration. We think synuclein spreading can be predicted from the connectome, and introducing synuclein to other seeds should cause this, a stereotypic type of spreading. So these are the next few experiments that I'm going to show you very quickly. So one of the ways we looked at synuclein spreading is by looking at a genetic model of clearance. And so, if you have too much synuclein in the brain, one of the ways we hope we can reverse neurodegeneration is by helping the clearance. And we looked at this protein called heat shock protein 90, HSP90, which, it took my PhD student Medini forever to explain this to me, so I'm going to try to explain it to you in a much simpler way. But basically what it does is that when it comes in, when it sees alpha-synuclein in its environment, it determines whether or not it's folded or misfolded. If it's folded, it lets it go. If it's misfolded, what it does is it degrades it and sends to the proteasome. Sends to the proteasome for degradation. And so in this case, but in the cases where you have too much synthesis and not enough clearance, it has a really hard time doing this degradation and sending misfolded alpha-synuclein to the proteasome. And this is because it gets stuck in this complex called the epichaperome, which is a really challenging name for me, but it can't do its job when it's stuck in this epichaperome. And so one of the ideas is that if you drive HSP90 expression, you can help tip this balance back to clearance instead of synthesis. And so we did that in a model that overexpresses HSP90, so overexpresses this protein that helps degrade misfolded alpha-synuclein. And the takeaway is that these animals who have been injected with preformed fibrils were overexpressing alpha-synuclein. When they also overexpress this HSP90, they reverse that phenotype, that behavioral phenotype that I showed you just moments earlier. That's, in this case, that's the pink line here. And this is work that we did with Marco Prado at Western University. And here I want you to focus in on a few things. So this is just, on the top is the unaffected animal. In the middle are the animals that I showed you that are injected, and that's the canonical neurodegenerative pattern that I showed you. And here, in red, what you see is a reversal of that neurodegenerative pattern if the mice are allowed to clear alpha-synuclein properly. And that helps us demonstrate that all this machinery, all this work that we're doing could potentially be used to understand therapeutic interventions or genetic targets for disease. Okay, so we got a check mark there. I'm going to go a bit over time. I apologize, but not that much. So one of the things that we did again, also introduced by Jake beautifully yesterday, if you were at that talk, is we tried to see if we could adapt tools that were developed for the human to work in our animal model work, animal model data. So work from Alan Dagger and Bratislav Mičić has shown that in the human PD, you can pretty well predict what the atrophy pattern should be based on the structural connectome of the human. But if you add into that model information about amyloid clearance, so basically amyloid gene, sorry, not amyloid clearance, synuclein clearance, and that's GBA gene expression, and synuclein synthesis, which is SNCA gene expression, you can improve the model fit quite substantially. That's what you see here on the plots on the right. And so in the mouse, this is somewhat possible as well, using transneuronal connectivity from the structural connectivity atlas, so the Allen Human Brain Atlas, and the gene expression data that exists in the Allen Brain Atlas as well. And we can model the rates of infection and neural death using similar machinery created by Alain and Bratislav. And when we do that, what we see, and what I want you to focus here on is this peak correlation, because it kind of estimates this in a pairwise fashion, is we get this peak correlation. We can predict the atrophy that we actually see in our animal model data with an R of 0.64, which I think is really, really good. So suggesting that, once again, this is actually predictable by the connectome. So we get a check mark there. So now, if we introduce synuclein into other seeds, this should also cause spreading, and we wanted to test that. So this is work done by Janice Park, who's sitting here. Very similar experimental paradigm, except this case, what we did is we interjected into that place where we didn't see any neurodegeneration, the hippocampus. And what we end up finding when we do the same type of analyses is that we get this very focal atrophy limited almost only to the hippocampus, and you almost see in certain parts of the hippocampus a 30% reduction in volume based on this injection of alpha-synuclein, but doesn't really propagate in the same way outside of the hippocampus, which was very curious to us. So it doesn't really follow the same patterns as we expected from this striatum. And this is true if we kind of put it through our computational model as well. And if we compare this to different papers that have done this type of injection and looked at spreading, they see spreading of alpha-synuclein to all sorts of different areas, whether you inject in the striatum, so some of the ones that we saw in our earlier experiments, or if you inject in the hippocampus. But it suggests that the spreading itself is not enough, and there's something about cell type specific vulnerability that might be really important when it comes to what happens in terms of the neurodegenerative properties. So this model, that's a computational model, which is very informative, is also incomplete and can also be informed by these types of experimental data, and we're hoping to kind of investigate that further, and we're very excited about that. So we get a question mark there. So I think what we've shown today, the conclusions are, we've demonstrated that the use of longitudinal imaging is a potential drug discovery tool for testing synuclein clearance, and we're actually using this methodology to test 3 different compounds at the moment. The prime hypothesis is true sometimes and can be predicted via computational models. And then we still have a lot to learn about this, and I think we can learn from these other types of bespoke experimental paradigms to improve our understanding of how synuclein propagates and causes neurodegeneration. So I'd just like to finish by thanking my lab, who are tireless and incredible, collaborators at the MNI in Western, and my son Pinaki, who is very mad about all the food pictures I'm sending him from here in Korea. And with that, I'd love to take your questions. Well, I'm not taking your questions. I'm going to thank these folks, and I'll take your questions at the end. I clearly left this slide over from a different presentation. All right, thank you very much.